And with this, it's my pleasure to just introduce my good friend and colleague, Antonio Zorzano. Antonio is full professor at the University of Barcelona. He's a basic scientist with a fantastic career and achievements. He's also a very nice person, very approachable. We always have a very good time when we meet. And I think that, Antonio, I'm not going to say very much. I mean, your biography is here. They can read it. And if you come here, I will pass the word to you. So welcome.
Thank you very much.
Welcome.
All right, so good morning to everyone. First of all, I would like to thank the organizers, CIBERDEM MSD, for this fantastic setup and for organizing these type of meetings, which I think are really very exciting for all of us, especially certainly for the people in CIBERDEM. And what I would like to talk to you today is about some of our most recent data dealing on aging and skeletal muscle. And our lab is essentially a diabetes lab, a basic diabetes lab. So, in fact, this analysis of aging, in fact, was not my idea. And we should blame on somebody who is right here, David Sebastian, who is right there. So he's the guy who's been responsible for the ideas and for the development of what I'm going to be explaining. So you all know that aging, together with obesity and physical inactivity, are major processes involving disease susceptibility in the developed world. There is a lot of recent work being done on aging and on obesity, of course, not so much on physical inactivity. But let's focus into aging. It is very clear that our global population is becoming older. Those are data taken from the World Health Organization and the predictions between the situation in 2015 and 2050. The colors you see here depend on the percentage of people in the population which is 60 years or older. And as you see, this color is getting more intense. It's going to be getting more intense, which essentially means that we are going to pass from 900 million people above 60 years of age in the 2015 to probably 2 million in the 2050. So those are really big numbers. If we go to the European Union state members, what we have seen in the last almost 60 years is an increase in almost 10 years in the life expectancy, both in women and in men. And this is going to continue. And those are the predictions from the European Union. So the numbers are really spectacular. So my first question, coming from the very basic scientist, is what do we think is aging? Right now there is a certain consensus indicating that aging represents a progressive accumulation of damage leading to tissue dysfunction. And the damage can be a consequence of cellular senescence, stem cell exhaustion, many different aspects. Altered intercellular communication, genomic instability, telomere attrition, epigenetic alterations, loss of proteostasis, not one, many different alterations. And of course, mitochondrial dysfunction, which is going to be something we are going to be focusing on. This is something it's not my point of view. In fact, it's the point of view of Carlos López-Otín, who is an expert in this area. And there are some additional thoughts that perhaps we need to consider. One is that aging is not caused by active gene reprogramming. It's not a consequence of an active gene reprogramming. And the second is, which became clear already, that there is not a single cause for aging in our population. Probably we can get that in animal models, but and probably in some conditions of progeria in humans, but this is not the normal cause, a single cause of aging. And the third is that somatic maintenance and repair functions, the ones that each organism has, this is optimized for the period of the organism is expected to survive. And that is something that is difficult to break. We are expecting to be a certain age for every type of organism we deal with. So the levels of maintenance and repair are appropriate for that, and not for just increasing twice as much or getting a huge increase in lifespan. So no wonder that based on this situation, aging is associated to many different diseases, and that goes from cancer, heart disease, to type 2 diabetes, many others. Okay, from the point of view of skeletal muscle, there are two aspects that I want to emphasize. One is the fact that in muscle, or muscle during aging, undergoes or suffers insulin resistance. This is responsible for glucose intolerance. And in addition, during aging, there is a reduction in muscle mass. There is sarcopenia. This leads to muscle weakness, and this is related very much to the frailty syndrome. And right now, and this is an extremely important aspect in disability and incapacity to maintain yourself. And there are no really treatments here up to this moment. So it's extremely important to focus into the potential stopping or reducing the rate of muscle loss during aging. Okay. What do we know in connection with mitochondria and aging in human muscle? What we know, probably there are many different data, first of all. But probably the very exciting ones came like 12 years ago by the NERS group, indicating that there is this reduction in the rate of mitochondrial ATP production with aging. And this is independent on what the substrates are provided. The mitochondrial substrates are provided. So that is the difference between these different panels, but I won't go into these details. But there is certainly a very marked reduction in the rate of ATP production during aging. A second aspect, at the more basic level, is that if we introduce lots of mitochondrial DNA mutations just by making transgenic mice that express a mutated form of in fact, of a Pol gamma A polymerase, what we get is a model of aging. This is data that were published 12 years ago by Niels Larsson's lab, indicating that you can generate aging as a consequence of an increased number of mitochondrial DNA mutations. In fact, during the last years, most of the data point to the following model. Initially, it was thought that mitochondrial DNA mutations were really relevant in every single cell type, but later on it became clear that somatic cells are not playing such an important role, and essentially this accumulation of mitochondrial DNA, in fact, has a major impact in stem cells. So as a result of enhanced or increased number of mitochondrial DNA mutations, there are alterations in the functioning of the stem cell. The stem cell has less capacity for cell renewal. There is stem cell exhaustion, and this relates to aging. So this is a model. Well, so there is a need for further demonstration of that. So it is very clear that one of the aspects that deal with aging is mitochondrial dysfunction. Now, let me tell you, mitochondria are not just those isolated organelles that I used to study when I was in school, but in fact, when you stain mitochondria in cells, this is what I'm showing you here. What we find is these beautiful filaments, this network, and those are mitochondria. And this is a result of a process of mitochondrial fusion and mitochondrial division. Certainly what you see here is more fusion than division. So if I could have the video, what you will see is just 10 minutes in the life of a cell. So this is a cell in which we have just stained mitochondria. This is a normal cell, nothing special about it. And then what you will see is that you will see that all these mitochondria, first of all, are not just sitting, they are really mobile, they are really dynamic, and you will find how these mitochondria are really fusing in some cases. So you will see two filaments that they are separated initially and they fuse, and vice versa. So you see that this is really moving, and the reason it's moving is because it's really linked to the cytoskeleton. But in fact, in this area and in this area, what you should see is that there are some mitochondria that are initially forming filaments and they break apart, so there is division, and you may, well, we will go through another one. And you may be seeing here that there are two filaments that they are fusing. So this is something that is not that strange. This occurs in every single cell. The exception is when this does not happen, okay? So this occurs in every single cell. This mitochondrial this dynamism and this balance between mitochondrial fusion and mitochondrial fission. Okay, so this process of mitochondrial fusion and mitochondrial fission is catalyzed by a number of proteins. And I want to just to focus into the proteins that are important in mitochondrial fusion, and those are these three proteins I'm pointing to you here. So one is mitofusin 2, mitofusin 1, and OPA1. And these proteins, mitofusin 2 and mitofusin 1, they are sitting on the outer mitochondrial membrane, and they tether to neighbor mitochondria, so they tether them, and after that, they initiate the process of fusion. And the fusion of mitochondria entail both the fusion of the outer mitochondrial membrane as well as of the inner mitochondrial membrane. And the fusion of the inner mitochondrial membrane is catalyzed by OPA1. Okay, so what I want to do in this talk is just to focus to this particular protein, mitofusin 2. It's one of the proteins that we are more excited in. So they are very similar to mitofusin 1. They contain a GTPase domain. They have two transmembrane domains at the outer mitochondrial membrane. And as I was telling you, they are really important in tethering neighbor mitochondria. They are also important for in human disease. And we know that there are mutations in mitofusin 2 gene that cause a rare disease, Charcot-Marie-Tooth type 2A neuropathy. Okay, and they are really important. There is no cure for it. So it's also, this has this very important angle. Okay, what happens if we, in the lab, we generate cells which either normal or they are ablated for mitofusin 2 or for mitofusin 1. I'm showing you here is the mitochondria. So what you see is the normal mitochondria, nice filaments in wild-type cells. And what you see here is that mitochondria are really forming fragments. They are really fragmented. So you see essentially points, big points, or small points. So these proteins are really playing a very important role in controlling mitochondrial morphology. You go from these networks to just these dots, or mainly dots, okay? So in mitofusin 2, is not just a protein that is important because it controls mitochondrial morphology. It is important because it controls the way mitochondria function, the way they operate, and this has very profound implications in metabolism. So I want to convince you very quickly that mitofusin 2 is a modulator of mitochondrial metabolism, but it's also important in glucose disposal, in insulin signaling, and in energy balance. And this has implications in humans. Okay, so we started this work just with this observation. What we found after cloning mitofusin 2 is that mitofusin 2 is repressed in muscle from obese subjects and also in muscle from type 2 diabetic people. So we wanted to analyze what were the implications of this reduction. When we work with cells with normal values of mitofusin 2 or with no mitofusin 2, what we find, as you say, this is not a surprise anymore, a big change in morphology. But together with this change in morphology, what we find is that these cells with no mitofusin 2, they produce more ROS. And in fact, they show most of the times we find oxidative stress. And under these conditions, more oxidative stress goes together with less mitochondrial respiration. And under these conditions, there is more mitochondrial calcium. And this is a general observation we find in every single cell, or even tissues, after ablation of mitofuscin 2. What is the impact under in vivo conditions? Here, what I'm showing you is data from liver-specific knockout mice. So mice are normal, with the exception of the liver. And those hepatocytes, the liver, don't express any more mitofuscin 2. What happens under those conditions? 2-month-old mice, which are really young mice, they show glucose intolerance. This is what I'm showing you in orange, when they are treated with a normal diet. And same thing happens when they are subjected to a high-fat diet. So they are really glucose intolerant. And under those conditions in which they are glucose intolerant, they undergo a transient hyperglycemia, and they show a very high hyperinsulinemia, especially under a high-fat diet, indicating that, suggesting that these mice are profoundly insulin resistant. And indeed, they are insulin resistant. And in fact, what we know, and we learned some years ago, is that the signaling pathway of insulin is really altered. And this is something we have found in liver, we have found in skeletal muscle. And for instance, what I'm showing right now is data in muscle in which you see how insulin stimulates the insulin receptor phosphorylation. That's perfectly normal. However, downstream events of the insulin receptor, such as the activation of PI3 kinase. This is the normal activation in normal muscles, and this is what happened in the knockout muscle. This is really altered. And same thing with downstream events as AKT phosphorylation. So insulin signal is deficient, the capacity to respond to insulin is deficient, glucose intolerance is deficient. This is telling you how important mitofuscin 2 is in liver or in muscle. But this role of mitofuscin 2 goes beyond liver or muscle. And in collaboration with Marc Claret, in fact, Marc Claret was really the senior author in this study. He is an extremely good neuroscientist. What he did is he ablated POMC neurons. He eliminated mitofuscin 2 in a very few number of neurons in the hypothalamus. This is the POMC neurons. These neurons are very important because they block food intake. And what happens when mitofuscin 2 was out is that these animals were obese, very quickly were obese. And this was a consequence of an enhanced food intake, which is something expected, as well as a reduction in energy expenditure. So this leads to obesity. The absence of mitofuscin 2 in a very few number of cells in the hypothalamus, but very few, but very important, the POMC neurons, this leads to obesity as a consequence of at least 2 different molecular effects that we need to characterize. What happens if, what is the role of mitofuscin 2 in adipose tissue? This has been done very recently in Carlos Cantó lab in Lausanne, and essentially the data also point out to a very important role of mitofuscin 2 in brown adipose tissue. You know that brown adipose tissue is essentially a thermogenic tissue, and what in fact Carlos has demonstrated very recently is that animals with no mitofuscin 2 in adipose tissue, they have difficulties in maintaining temperature when animals are subjected to cold. And in fact, they respond very badly to a beta-3 adrenergic agonist, indicating that the brown adipose tissue is not really working properly. So all this data suggests that mitofuscin 2 ablation impairs brown adipose tissue thermogenic function. Some of you may be saying, well, yes, all this is okay, but all this is mouse data. Okay. But what we know, and this comes from a very recent publication in eLife by Robert Semple's group, is that they found 4 patients with human biallelic mitofuscin 2 mutations. And these mutations in the 2 alleles, that is characterized by upper body adipose hyperplasia and a very marked suppression in leptin expression. So we do think that mitofusin 2 is going to be playing a very important role, not just in mouse health or disease, but also in metabolic disorders in humans. So this is our scheme for mitofusin 2. We think that mitofusin 2 integrates different signals, integrates pro-inflammatory factors, dietary fat, exercise, exposure to cold, and mitofusin 2 senses all these signals and leads to an integration which is converted into a modulation of insulin signaling, and this is something very clearly demonstrated in liver and muscle, but also in adiposity, based on the effects in adipose tissue as well as in hypothalamus. Okay, but I haven't told you anything about aging, right? So what I want to tell you, essentially, I want to convince you is that at least in mouse models, mitofusin 2 is involved in some of the muscle alterations associated to aging. And as I was saying before, everything started when David Sebastian, was willing to analyze the impact of aging. And I said, well, I don't know whether we will be able to pay the bill. But he convinced me to do so. And the data were really promising. Initially, when he was comparing 6-month-old mice and 22-month-old mice, which are really old mice, he showed a very substantial, very profound reduction in mitofusin 2 protein in skeletal muscles, in different skeletal muscles. And this was progressive. So when he was doing 6 months, 12 months, 22 months, there was a progressive reduction in mitofusin 2 expression. Okay, so under those conditions, what he did is he analyzed the impact of ablating, of eliminating mitofusin 2 just in muscle on the capacity to age, or how these animals were aging. And here you have some data. I won't show everything. Just this provides you an idea of how much oxygen consumption is being used by mice during the day, during the daytime, during the nighttime. And this is in young mice, control and knockouts, and same thing in all control and knockouts. When we integrate all this data and we quantify that, what we find is that with aging, there is a reduction in oxygen consumption. This is already known, okay? This is very well known. This is daytime, this is nighttime, okay? There is a nice reduction. But notice that in mitofusin 2 knockout mice, this reduction is even bigger, indicating that these animals are really having a worse aging than normal wild-type mice. What happens now if we take a look at the capacity of these mice to use glucose? Well, this took a long time to analyze, because when we measured initially, when David measured initially a glucose utilization in 6-month-old mice, he found nothing. And under those conditions, when he was taking a look at plasma insulin levels, which gives you an idea of what the level of insulin sensitivity, he found no changes. When he did the same experiments in 12-month-old mice, again, no changes in glucose tolerance. But under those conditions, look at how insulin levels were. They were extremely high. And high levels of insulin are really suggesting you that there is insulin resistance under those conditions. And in keeping with this, what he found was an incapacity to respond to insulin by reducing glucose levels. So these animals at 1 year of age, they were insulin resistant, and they show incapacity to respond maximally to insulin. And what happened if we extended these studies to 22 months of age? Under those conditions, what we found was glucose intolerance, insulin resistance, and again, in reduced capacity to respond to insulin. So these animals with no mitofusin 2 in muscle, they were again showing a worse aging process. What happened when we took a look at muscle histology or muscle force? Well, that was very surprising. What we found was that these mice with no mitofusin 2 in muscle, they were really atrophic, Under those conditions, they did poorly when they were forced to run in a treadmill. So look at how bad they did. And in fact, they show lower grip strength, and they show lower normalized maximal tetanic force. So these muscles were not doing well. This is an example in mice of unhealthy aging. Okay, and what else? What we found is that under those conditions, these mitochondria were not working well. And as a result, there was oxidative stress. Look at this is protein carbonylation, which is a pretty good parameter estimating oxidative stress. These animals, they really show oxidative stress. And how was that? Our initial electron microscopy observations were really suggesting that under those conditions, we were having weird mitochondria. So because of this, and because of some connections between mitofusing 2 and autophagy, we analyzed what was going on with autophagy. So first of all, what is autophagy? Autophagy is a complex process which has as an objective to collect in an isolation membrane organelles or proteins which are damaged. They form an autophagosome, a double membrane containing all this damaged material inside, and this material is fused to lysosomes, and this gets degraded. So under normal conditions, this is a fantastic way to get rid of damaged organelles or damaged mitochondria. And this system is complementary to the proteasome. Okay? So this is an extremely complex process, and there are many factors. In one moment in time, we thought that perhaps autophagy was altered under conditions in which mitofusing 2 was down. So what we did is we follow a very common procedure. Because this is a continuous process, and this is formed, so there is formation of autophagosomes, there is fusion with lysosomes, and there is degradation. What we do is we analyze the rate at which this takes place. How do we do it? It's very simple. What we do is we block with different compounds at this level, more or less. So what we do is we measure the rate at which these different autophagic proteins are built up with time. And this is what I'm showing you here. So what I'm showing you is how control cells in white, they respond to blockage of autophagy, and you see how these different proteins, some of them, this one, for instance, is a mitophagy protein, how it builds up. And look at what happens when these cells are really knocked out or knocked down for mitofusing 2. There's no build-up anymore. And this is really suggesting to you that under these conditions, there is a defect in the rate of mitochondrial degradation via autophagy. So these and many other data that I don't have time to analyze, they permit us to suggest that mitofusing 2 is a very important protein, at least in skeletal muscle, and links mitochondrial fitness with healthy aging. We need mitofusing 2, not just to maintain mitochondrial respiration, not just to maintain the capacity to respond to insulin, not just to maintain energy expenditure, also to maintain muscle atrophy, muscle mass, an appropriate muscle strength. When this doesn't happen as a consequence of alterations in mitochondria and also alterations in the capacity to degrade this mitochondria, this damaged mitochondria, muscle atrophy occurs. And this is something we want to avoid. So this is essentially our model these days. Just to finish, I would like just to mention a few aspects. One is that as a result of this situation in which there is, we could say, a mouse unhealthy aging, what we have been able to find is that there is induction of an adaptive pathway, which may be also something to explore if we want to, in fact, look for a strategic of pharmacological strategies. So what we find is that under conditions in which mitofusing 2 is out, what we find is an induction of HIF1 alpha. This means pseudo-hypoxia. this is extremely important, this HIF-1 induction, because when we block it with a chemical compound, the NSC compound, what we find is that HIF-1 alpha is really lower, but under those conditions, what we find is that there is no more induction of mitophagy genes. And in fact, what happens under those conditions, when we block this capacity to overreact, and the muscle overreacts to the loss of mitofusin 2. When that doesn't happen, there is even more oxidative stress, and there is a failure for mitochondrial ATP synthesis. So based on that, this is the model we have proposed, and which is the following. In a young muscle, normal muscle, healthy muscle, mitofusin 2 plays a very important role participating in mitochondrial quality control. In a normal aging, mitofusin 2 goes down. And this reduction reduces mitochondrial quality control, and as a result, there is more accumulation of mitochondria. This is an important aspect. This is very important. And this triggers a pathway, which I didn't mention, but we know that is driven by ROS. And ROS induces HIF-1 alpha, and this induces a number of mitophagic proteins which try to balance this mitochondrial quality control. However, when mitofusin 2 is really down, no mitofusin 2, this process is even worse. Mitochondrial quality control is really altered, it's highly compromised, there is more accumulation of damaged mitochondria, and this triggers a pathway which can no longer provide this compensation. So under this condition, pathology, sarcopenia occurs. So those are the conclusions of my talk. One is that mitofusin 2 is dysregulated in obesity and type 2 diabetes, and its loss of function alters mitochondrial metabolism. We think that is a good drug target for metabolic diseases. Mitofusin 2 modulates glucose disposal, insulin signaling, and energy balance through its effects in muscle, liver, adipose tissue, and also hypothalamus. Mitofusin 2 is involved in some muscle alterations associated with aging, such as insulin resistance and atrophy, very important, this aspect. And mitofusin 2 links mitochondrial fitness with healthy aging. It represents a good target, not just for metabolic diseases, but also for the treatment of metabolic disorders associated to aging. This is our hypothesis. And finally, I would like to mention that I haven't done anything of what I'm show you today, and especially the champions today were David Sebastian, who I mentioned already, many other people working with mitofusin 2, and also I want to mention Andrea Irazoki, who may be here also. Yes, you are. And she did her master last year, now doing the PhD, and she did very good work also finishing some critical aspects on this project. And I also want to mention the collaboration with many colleagues and friends here for this talk, essentially Carlos Cantó at the Nestlé Institute of Health Sciences in Lausanne, and Marc Claret at the IDIBAPS. So thank you very much for your attention. Thank you.